everyone. Today we're discussing DNA technology. So within DNA technology, there are several different methods for which you can study genetics. And so when you think about the DNA within a cell, it's housed in the nucleus. And so the first step of any procedure that you do in the lab is you want to actually take out or extract that DNA, but you don't want anything else along with it, like proteins or RNA material or lipids or anything else getting in the way. And so extracting DNA is the first step in any biological technique. And so this is a picture on this slide as to how you may go through extracting DNA. And so the first step of that process is using a detergent that will help disrupt the cell membrane. So that helps to break it down such that the DNA can escape from the cell itself. Next, you want to destroy anything else that could interfere with the process of isolating the DNA. So like a protease to destroy proteins or an RNase to destroy RNAs. You want to make sure you get rid of all that extra material so you're just left with DNA at the end. Now, you use a specific type of instrument in the lab called a centrifuge. And a centrifuge essentially spins your tube sample at a very high rate in order to pellet all the material at the very bottom. So that's again what's known as a pellet when it comes out of the centrifuge. The supernatant is the liquid portion that remains above the pellet. And essentially what you're going to pellet down versus what you're going to suspend in liquid that comes out of the centrifuge will be your DNA material that you will transfer to a new tube. So the rest of the cell debris that you don't want, like lipids and phospholipids and maybe proteins that were not destroyed yet, those are all pelleted in this little pink area here. And then the supernatant, the liquid portion, is suspended right on top. And it's a very tiny volume of liquid, maybe 100 microliters or so. And it's very difficult to try to extract that away from the pellet such that you don't get any of that residue from the cell in your DNA sample. And so then you transfer that to a brand new tube and you can precipitate the DNA out with alcohol. So ethanol is used and then the DNA would be suspended in that ethanol because it brings it out of the solution. So this is one way, and there are different kits that you can buy through various DNA companies to take out and isolate DNA without taking out anything else that could interfere with the DNA. At that point, you would buy a kit from a company to purify the DNA. Make sure it is essentially clean of any other residue. And that would make the DNA workable in the lab through various other methods in order to extract meaningful information from the DNA. So nowadays, people do this in preparation for taking out DNA and perhaps sequencing it. So figuring out the letters of A, C, T, and G. Now, we actually have 3 billion bases of DNA that makes up the 46 chromosomes that we have. And to sequence all 3 billion bases has come down in cost from millions of dollars to just $1,000 for one person. So that's not too extreme based on the fact that we've only been in sequencing a few years. So 10, 20 years or so, the best sequencers have been around since then. And so now we can get an accurate DNA read by figuring out those 3 billion letters of DNA and how they're arranged. The Human Genome Project did this back in 2001. And so if you think about how far we've come, it's been almost 20 years or so. And so the DNA sequencing technology has really made it feasible for every human to eventually have their full DNA sequence that tells them everything in their DNA blueprint. So this would be extraction and isolation. If you buy a kit from a company like 23andMe or Ancestry, they send you a kit with a little cotton swab that you use to swab the inside of your cheek. So it suspends your cheek cells. Then you send that back to the company and they will sequence a few genes, like maybe 100 out of 25,000, and they'll send some information back to you on your health and your ancestry based on those 100 genes or so. And so because of that, they will receive the kit back with your sample and they will go through the process of extracting that DNA and purifying the DNA. And then they will sequence it, so figuring out those letters. Now, in preparation for sequencing the DNA, you first have to amplify it because when you have that resulting DNA here on the right-hand side, that's actually all of your DNA, all 3 billion bases. Sometimes in a project, you only want to look at one gene. And because of that, you need to pinpoint in a sample where that gene is. So you actually need to copy that gene a billion times in your sample so it stands out. So when you go through sequencing it, for example, it's only sequencing that specific gene. So many researchers are interested in single gene disorders or single gene mutations, and hence they go through this process called PCR, polymerase chain reaction, which all it is used for is copying the DNA. It's essentially DNA replication based on heating and cooling cycles in a little thermocycler machine. So you already know replication. Double-stranded DNA opens up. And so in that little machine, a high temperature is applied to actually open up those DNA strands. And then a process called annealing happens, where little primers, little RNA pieces are added into the mix. And then the enzyme, a type of DNA polymerase, comes in and it'll start adding nucleotides to both strands. Essentially, this is exactly the same as the process of DNA replication. And what happens is, at first it's heated to open those strands, but once you add that primer and the polymerase to add the new nucleotides to both strands, you need to actually cool it. So there are several heating and cooling cycles. Once you have the two new daughter DNA molecules, you start the next cycle. You heat it again, then you cool it to anneal. Then you heat it again to denature, and then you cool it to anneal and extend. And so these cycles continue 30 to 60 times, and after just an hour or two, you have a billion samples of that single gene that you're interested in. And so this really helps for studying different diseases. Now, before you sequence it, you might want to quality control check that the PCR worked. And a gel, and this is a type of polyacrylamide gel shown here. It's actually the image of the gel. But this little gel is formed from a jelly-like material. And what happens is this little gel, it's very small. It sits in a little container that contains some buffer, some aqueous liquid, and then an electric current is applied to the gel. And you essentially place your DNA sample onto it in these little slots called wells. And see, this is the top of a well here, and you can apply your sample in here, and it will run down this particular lane of the gel. And wherever your sample shows one of these little bands that you see in these various wells, that shows you that yes, there is a DNA product there. So yes, you did the PCR correctly. Sometimes you might be looking at one gene, sometimes two fragments of a gene, and so that's why you see these various banding patterns here. This gel, when it comes out, is actually looked at under a special type of visualization called ethidium bromide staining. And so that's why you see the coloring of this gel. It's black and it has these very white signatures coming off of it, and that's the staining with ethidium bromide. Sometimes gels will be stained different colors, so you would see different results in different literature. So this is purely used for visualizing your product. It's a quality control check. Now, in many types of labs, you may not know what your gene necessarily does, but you may be interested in cloning it in order to figure out what it does, or to see if you can get it um, in a new animal or a new species. So this is an example of molecular cloning, and all that means is you're copying the gene. So this is another way of copying a gene, but you're doing it in an organism itself. And so we're looking at a gene, in this case, over here on the right-hand side, that allows jellyfish, in this case, to glow green. And so we have a whole bunch of foreign DNA here. This is from jellyfish, and we know that jellyfish glow green, and we know that that gene is somewhere in this foreign DNA. So what we want to do is figure out where, and figure out if we can put it in another organism. So we take what's called a plasmid, and a plasmid is a commercially made circular piece of DNA that contains a marker gene like antibiotic resistance, but it also contains the gene that we are interested in. So let's say the area of the gene that we're interested in putting in here would be the fluorescence gene from jellyfish. So what you can do is you can cut open this plasmid, you can cut open this foreign piece of DNA into smaller sections, and then you can insert it into a whole bunch of different plasmids. And then you can place the plasmid material in bacteria. And bacteria are good to use because you can easily visualize whether that gene is active, because the bacteria will then glow green. And so the bacteria that received the proper fragment of DNA that had that green fluorescence protein on it would be the one that now carries that piece of DNA. Then you could take that bacteria, you could sequence it, and then you can figure out the sequence of a plasmid, which would then contain that piece of DNA that you received. And so this is one way through molecular cloning that you can achieve the result of transforming a gene from one organism and growing it in another one. And you can continually grow it in another one to produce more and more of that gene. So those individuals who work on production of special proteins like insulin, this is the kind of methodology that they would use. They wouldn't grow it in bacteria necessarily. They might grow it in a plant, and then the plant produces that special protein. So once we know what the gene is that codes for the special protein we want to see, we can clone it. Now, reproductive cloning is another biotechnology method, and this is an example here of reproductive cloning of a sheep called Dolly. That was the first animal to be cloned, and it's a process called nuclear transplantation. So essentially what you take is you take a sheep and you take out a cell from that sheep, and this is called your cytoplasmic donor. So we know within a cell we have a nucleus. So from the donor we can actually take out the nucleus, and then from a secondary donor, which is the nuclear donor, we can take out mammary cells from that individual, take out the nucleus, and then place it into the cytoplasm of the first donor, the cytoplasmic donor. So basically, we want to make an exact clone of this sheep up here, the nuclear donor. And so we grow this individual in a little petri dish. It starts to develop into a blastocyst, which is an early embryo, and then we get a surrogate sheep and we implant that early embryo into it. And so because this embryo is growing based on the nucleus of the nuclear donor, therefore, the growing sheep, which is called Dolly, that's born, should be a genetically identical copy of that nuclear donor. Now, from my lecture last time, you would have heard me discuss the idea of epigenetics. So, is Dolly truly a 100% genetic copy of the nuclear donor? No, because epigenetic factors, so being in the womb of her surrogate mother here, could have altered some of the gene expression of her genes. So, such that she might have a few different genes that are activated in a different way from her donor. But initially, when this process was completed, it was thought that Dolly was 100% a genetic clone of the nuclear donor, which we now know would not be an exact clone. Now, the most promising results from all of these initial biotechnology methods is that you can essentially engineer organisms how you want. And the most exciting breakthrough is a technique called CRISPR, CRISPR, all capitalized. And this is the way by which genetic engineering can be accomplished. So essentially, if I find a gene that's mutated in a patient, I could just introduce a new good version of that gene, so a non-mutated form, into that patient to cure them of the disease. And this is how CRISPR is working through this method. Essentially, what you have in gene therapy is you have a vector. In this case, a virus can be used as a vector, a virus that is not activated. So it's been stripped of its own nucleic acid. This virus, I can put a gene that I want into it along with another machinery to guide it to the proper place in the genome. And I can put this into a person or a cell, and I can allow that virus to make its way into the nucleus and actually insert the good gene into the DNA of the diseased individual or the diseased cell and cut out the bad piece. So it's exciting that we now have machinery that utilizes this function, whereby which we can take a good gene, introduce it into a patient such that the bad gene might be taken away. The bad gene in this case signifying a mutated sequence that leads to a disease. You can do the opposite as well. So if you want to insert a mutation into a patient, you can as well take your virus, insert the machinery that's used to make cuts in the DNA, package it in, and the virus will make its way into the cell, into the appropriate location, and will cut that gene that you want cut. Sometimes by making a mutation, we actually make a person healthier. So there are some individuals whereby they have mutations in specific genes that allow them to become resistant to disease. The most exciting of which is a mutation in the CCR5 gene, which allows a person to become resistant to getting HIV. And so that's what's in clinical trials now, is using this technology CRISPR to take a virus that's not working, to insert the machinery used to make a cut in the DNA. That makes its way into the person. It makes its way and finds the CCR5 gene, makes a cut or a mutation in it, and then that person would be resistant to HIV. So that's what's in clinical trials, and it's really exciting right now with the advent of CRISPR technology. So essentially, you would be able to then essentially engineer individuals with either healthy genes or taking out diseased genes, and really this type of technology could be applied to any gene in the genome. What's something that still needs to be looked at are the side effects. What are the side effects of having this type of technology or guiding system in the body, because originally it comes from bacteria, this technique? And what are the lifelong effects of altering genes in this way? So those aspects still need to be worked out. And how do we apply our knowledge of biotechnology and genomics? That means studying all of the genes in the genome. We can know our personal risks for diseases and how we might be able to then offset that, how we might be able to change our lifestyle in order to decrease our risk for diseases. Pharmacogenomics, so now you can essentially tailor drugs to individuals who are affected by certain diseases. So there's a commonly used blood thinner called warfarin, and that instead of trying to figure out what the appropriate dose is for a patient based on the genetic code, you might know exactly what the appropriate dose is, and you don't have to go through that trial and error. Microbial genetics, so looking at the entire set of microbes within the human body. Millions and millions of microbes exist with us, and there are projects around the world whereby which researchers are trying to figure out what the DNA sequences are of all of those microbes, creating new biofuels by manipulating bacterial genomes, looking at ancestry using mitochondrial genetics. So, our mitochondria have DNA in them too. And we don't know a lot about why, but we do know that we can trace our ancestry through mitochondrial DNA mapping. And so there are projects on the way for that. And agriculture, meaning crop breeding. So we can breed crops to have certain traits that we want. We can also breed certain organisms or animals where we see desirable traits in them as well, and that's already on the rise. So there are many applications of genomics like this. Something you want to consider for each of these might be what are some advantages, but also might be some underlying disadvantages of being able to manipulate humans and other genomes in this way. So thank you for paying attention to this lecture. I hope you enjoyed it, and please let me know if you have any questions.